to do when carrying out aseptic techniques, because we've already learned the different types of aseptic techniques. From yesterday, we learned about heat sterilization. We learned about, you know, different types of techniques that involve filtration. So these are those things that you would need to do when carrying out these aseptic techniques. So washing hands is very important at the start of any lab. So this is important to remove any bacteria or dead cells that may contaminate your culture. Closing windows and doors, because these when you keep the windows and the doors open, it may actually create a draft, and that can probably have like an info on your results that you're doing in the lab. Sterilize bench with at least 70% alcohol. This is to remove any microorganisms and to create a sterile environment in your workspace. Wear gloves. Disinfecting the bench top. Repeated that. Keeping plates closed when not in use, because when you're inoculating or when you're moving from when you're inoculating and you're using your plates and your agar medium, you want to make sure that you don't leave the plates open too long, or even leave the caps open too long, because you don't want them to be exposed to any microbes that might be in the air or anything like that. And it's always important to always sterilize your loops and needles. So I remember yesterday they were doing a streak plate on the agar, and this is how it would look like after you're doing your streak plate. So this is what we're calling streak plating. So that's I think somebody said that they didn't understand what was being done yesterday. So basically what they were doing was that she was using the inoculating needle, and she was streaking like lines of the bacteria. Usually we do streak plating when we're trying to like isolate like a certain type of bacteria, because other times bacteria, they'll grow in like clumps together. Like example, you're trying to test maybe your skin, you swab your skin to try to find like, maybe you're trying to find if staphylococcus, if you can find staphylococcus on your skin, which would be, and maybe when you do you grow it up in some medium, but then you might find that there are more than, it's not just staphylococcus that you find on your skin, there are other clumps of bacteria. So we do like a streak plate in order to like isolate the medium, and I'll show you guys a streak plate video later on so you guys can understand how we actually do this. So biosafety levels. A biosafety level is the level of biocontainment precautions required to isolate dangerous biological agents in an enclosed facility. So biosafety levels go up from 1 to 4, with 4 being the most dangerous and 1 being the least dangerous. So the biosafety level 1, microbes are not known to cause disease in healthy hosts and pose minimal to workers and environment. And these are the microbes that you'll be using in your lab when you go to university during research. So these tend to be like non-pathogenic strains of E. coli. So the BSL-2 microbes are typically indigenous and are associated with diseases of varying severity. They pose moderate risk, such as staphylococcus. And the BSL-3, these microbes are more indigenous and exotic and cause serious or potentially lethal diseases, such as our Microbacterium. And then you have the BSL level 4. So these microbes are usually contained at very high level containment. They're transmitted by their aerosol, and they're frequently fatal without treatment. Few labs are actually at this level. So Ebola and Marburg, and even I guess your COVID-19 could be classified as a BSL-4. All right, so we're going to go, before I show you guys the videos, I'm just going to quickly recap what we did yesterday for Jazz, who was not here. So we're going to quickly recap what was done yesterday. I'll be uploading the slides later on, and the video will follow shortly. It usually takes us a while to get them cut, guys. So everybody's okay. Any questions that you want to ask? Anything that you guys weren't clear on, so I could just make sure to pay keen attention to when I'm going back over. Anything you guys were not clear on, persons who were here yesterday?
Not that I can think of, but if I see it while you're going through, I'll tell you.
Okay, all right. So, hold on, just give me a moment. Okay. All right. So, okay, let's continue. So, aseptic techniques. We said that aseptic techniques refer to a procedure that is performed under sterile conditions, and these are procedures to prevent culture, sterile media, stocks, and other solutions from being contaminated by the unwanted organism. Because if we're working with E. coli, we're working with staphylococcus, we don't want it to be contaminated with any microbe that might be in the air, any microbe that might be on our skin. That's why it's very important that we wash our hands and we, create a sterile environment. So why are aseptic techniques important? As I said earlier yesterday, I said that prevention of contamination of cultures from foreign bacteria in the environment. And you also want to prevent contamination of yourself and the environment with the microorganisms. But also these microorganisms, although at your level, BSL1, you'll be working with, they're not going to be deadly or anything like that, but they can cause you to get a little sick. So you might actually have like a little diarrhea for the weekend or something like that. So that's why it's very important that you guys learn these aseptic techniques. Sterilization. So sterilization refers to any other process that effectively kills or eliminates transmissible agents such as fungi, bacteria, viruses from surface, equipment, article, food, or medication, or biological culture medium. So disinfection is cleaning an article of some or all pathogenic organisms which may cause infection. So we have the three main methods of sterilization and disinfection, and these aren't the only methods, guys. These are just the ones I want you guys to focus on. You have gas sterilization, pasteurization, which is actually kind of a subset of the moist heat, which comes under almost like the autoclave. But these are just the ones I want you guys to focus on. So as we're saying, sterilization using heat is actually the most commonly used method and involves destruction of enzymes on other essential cell constituents. So dry heat is used from a temperature of 160 degrees Celsius to about 1,800. And this is about for 2 to 3 hours. It's used for glassware, metal objects, oils, and powders. Moist heat, which is actually at a lower temperature, 100, it's about mostly 121 degrees Celsius. And it's usually about 15 to 20 minutes, and it's used for saturated heat under pressure to kill microorganisms and spores. And it's used to sterilize surgical equipment and also the culture medium. So I'm not sure, I think what the culture, I know some of you guys not sure what the culture medium is. It's just basically what you use to grow the bacteria in, so it'll have a lot of nutrients, peptones, amino acids, what are needed for the bacteria to grow. You have different types of mediums as well. Some of them are selective, like if you're trying to find a certain microorganism, you might what's the word I'm trying to think of, you might actually like inhibit certain nutrients. So if you know like, or you might make certain nutrients of a higher concentration. So if you know like a certain organism those best or thrives in certain environment, like we know that if you're trying to look for staphylococcus, they tend to like salty, the salt concentrate agar medium. So you know that if you're growing a bacteria and it grows well on on a salty medium, you can probably infer that more than like it's staphylococcus. So heat sterilization. So dry heat is able to penetrate substances more slowly than moist heat, requires longer exposure time and higher temperature. The dry heat damages most of it by oxidizing the molecules, and that's how it denatures the enzymes, and the essential cell constituents are destroyed and the organism dies. Temperature is maintained for almost an hour to kill the most difficult of resistant spores. And moist heat is the form of saturated steam under pressure. It's mostly used and the most dependable method. Steam sterilization is non-toxic, inexpensive, and rapidly microbial and sporicidal. It rapidly heats and penetrates fabrics. Moist heat sterilization uses the autoclave commonly for sterilization and biohazardous trash. So the autoclave is the most commonly used method for the moist heat sterilization. And for the dry heat, it would be the oven and the fire and the Bunsen burner, which we use for flaming. So on your left would be the autoclave, the middle obviously would Bunsen burner, and the microwave. So we use the Bunsen burner when we're flaming, and the Bunsen burner also helps to create a sterile environment. So it's always important when we're using in the Bunsen burner, we're inoculating, we're using it to work as close to the flame as possible. Okay. So filter sterilization. So filters remove microorganisms from air or liquids. That filters include the HEPA filters are used to remove microorganisms and other contaminants from liquid or air. The membrane filters are used for sterilization of heat-sensitive liquids, and nucleation filters are used to isolate specimens for electron microscopy. Membrane filters are generally 0.22 to 0.45 micrometer in size and made of nylon or nitrocellulose. So radiation sterilization. So radiation relies on ionizing radiation, primarily gamma, X-ray, or electron radiation to deactivate microorganisms such as bacteria, fungi, viruses, and spores, and even parasites as well. Certain parasites such as Giardia, which you guys will learn about, are they're not immune to radiation, so it can be used to kill their spores and also any other cells that they produce. So due to the numerous advantages over heat or chemical-based sterilization techniques, this method is particularly attractive in medicine and healthcare fields. The UV radiation or ultraviolet uses a wavelength of 200 nanometers to kill microorganisms, and it destroys the basis of nucleic acids and inactivates viruses. Use is limited or useful for sterilization of surfaces and transparent objects. And it's not just, I mentioned yesterday that this isn't just used for just sterilization of surfaces. Places also use UV radiation also to disinfect water. So that is also something. So as I mentioned, use it to disinfect it from possible bacteria or parasites as well. So gamma is the most popular form of radiation, and this one actually uses an isotope called cobalt-60. They are very penetrating and commonly used for sterilization of disposable medical equipment such as syringes, needles, cannulas, and IV sets. Requires bulky shielding for the safety of operators, and they also require storage of the radioisotope. So aseptic techniques that we use are the flaming. So this involves using inoculating lube, and you pass it through the flame to remove any microorganisms that may be on it or cells. Culture transfer, that's when we're moving when we're this is also involves using the inoculating loop. We're probably trying to move from one culture. It's also maybe if we're using dilution as well, picking colonies, street plating, making sterile agar plates, pour plating, spread plating, and also when we're cleaning up, we have to use probably about 70% alcohol. So this is an example of how we do like a transfer. So you from the inoculating loop. So the inoculating loop is heated until it's red hot, and we usually pass so we start at the top of the inoculating loop and we'll bring it through the fire and then the organisms in the culture are dispersed by shaking. Also, you actually need to cool the inoculating loop before using it because you don't want to essentially kill the organism that you're working with. So the tube is removed and it is flamed in order to remove any organisms that might be on the tube cap, and also flaming helps to create convection current, which reduces which reduces the chance of infection or anything like that. So the loop full of microorganisms is removed from the tube and then it's flamed again and you close the tube. And also to make so you guys should know that you should never put the tube that you're taking, the cap of the tube should never be placed on the ground. So you have to be manipulating both the inoculating loop and the tube as well as the cap in your hand at the same time. It's a bit tricky when you first start it, but you'll eventually learn how to do it. Okay, so any questions before we go on to the videos I said I wanted to show you guys? Anything you guys are unclear about? No, miss. Okay, so Danielle says she's okay, so all right. Let me just all right, so stop share for a minute. Okay, so this video is aseptic techniques and street plates. You guys remember earlier I showed you guys some colorful looking plates, some round things, and you saw like some streaks. So this is, I'm going to show you guys how it's done. Okay, so I'm going to show you guys how it's done and the procedures and how we go about creating a sterile environment. Hold on, let me just, somebody ping me. Sorry guys, let me turn up my volume. I probably let me make sure I'm actually sharing my computer sound. Yeah, that was not on. It's about 16 minutes, but I don't think I'm going to make you guys watch the whole thing.
Welcome to the laboratory. This video demonstrates two key techniques in microbiology, aseptic technique and the street plate technique. Aseptic technique basically means to create and maintain a clean, sterile environment for you to work in. This is extremely important when working with microorganisms because you want to be sure that the organism you're transferring to the plate or a broth is your organism of interest without any contaminants. So to create a sterile environment, we first...
So that's what she's saying. So that's how we always wash our hands, because I was saying before, our hands actually have a lot of microorganisms on them, especially Staphylococcus. And if we're working with E. coli, we don't want them to mix up. Or even our dead cells on our hands can even serve as contaminants. So it's very important that we wash our hands before starting and even wear gloves if necessary.
Firstly need to wipe down the surface that we're working on. So you should have some bench cleaner. So spray the bench with the bench cleaner. Bench cleaner is basically disinfectant, which will remove...
Okay, and the bench cleaner, guys, is usually about 60 to 70% alcohol that we usually use. Don't make anybody tell you that we use like, or it's usually bleach or anything like that. Don't let anybody tell you that we use full bleach or anything like that. We usually have to add water or something. So it's never a full concentration of anything.
Remove any dust or unwanted organisms that may be on the bench. So wipe down the disinfectant with a tissue or some paper towel provided and place that in the bin. A key piece of equipment that you need to use to create a sterile environment is the Bunsen burner. When the Bunsen is lit, the flame creates an area of sterility in the air and also keeps the bench nice and clean as well. So you want to light that soon after you've wiped your bench down. So grab your matches, strike your match, and then ensure that the hole of the Bunsen is closed, and then you can turn on your tap. You should hear the gas coming out, and then you can light your flame, and your match can be disposed of in the pipette bucket. So when you're not directly using the flame, you should have it on the orange flame, just so that you're aware that it's there and that you don't accidentally wave your hand or arm through it. So the technique that I'm going to demonstrate is the street plate technique. So this involves transferring an organism, whether it be from a solid medium or from a broth culture, onto another solid medium. So in this example, I'm going to use nutrient agar, and I'm transferring the organism E. coli. One of the first things you need to do when performing the street plate technique is to actually label your plate. So you should have a permanent marker on your bench, and you need to label your plate with some key information. Because this is a
Also, let me just make you guys know that when we're after we're done and we inoculate or whatever we're doing with the organisms, when we're storing them, we usually store them about at 37 degrees Celsius, and we always, say example, we always turn over the plates when they're being stored in the fridge or wherever, because we want to prevent condensation. Because anytime a condensation goes on the plate or on the organ, they will actually mess up with the results that you get. So it's very important to turn over the plate from the agar side that it was on, and also to store it at 36. Most times it'll be at 37 degrees Celsius. Your teacher will tell you.
nutrient agar plate. I'm going to label the plate with NA, the abbreviation from nutrient agar, and you should also include your initials from your first name, your middle name and your last name. And in this example, I'm transferring E. coli. So you can write the organism name, or you could write the exercise number that this plate refers to. You must also include the date in your labelling. So you should always label your plate on the bottom of the plate, just in case the lid is lost in transit or is removed from the plate. So you would always label it on the bottom, and you also need to label it on the edge or the circumference of the plate, so it doesn't obstruct the view of what is growing on your plate. The street plate technique is actually shown in a diagram in your manual. So this is figure 1. To perform the street plate technique, it requires you to use a sterile loop and to transfer some bacteria or yeast onto a plate, and then to continue streaking it. So basically the purpose of the street plate technique is to dilute the number of cells, so that when you get to your quaternary lines, you should have individual colonies. So one viable cell from a bacteria or yeast usually gives rise to one colony. So in this diagram, you can see in the quaternary steps that there are streak lines which don't intersect with the tertiary lines. That is one way of performing the street plate technique. With your quaternary lines, you could also perform a vertical line and a squiggle in the middle of the plate. There are a number of ways that you can do this, and both ways are acceptable. So to transfer your organism, you want to make sure that your Bunsen flame is turned to the blue flame, so that you can see the apex of the flame. You will need your loop, and you're going to sterilise it in the flame. So basically the loop is a piece of wire with a loop on the end, and you want to make sure that the entire surface of the loop, including the stem, is sterile. So you basically hold it in the blue flame, and you can see it turns orange when it gets hot. And you want to make sure the stem and the loop is sterile. Once that's sterilised, you need to let it cool for a few seconds, so just hold it near your Bunsen flame. A way of determining whether the loop is cool enough is to actually touch it to the agar surface of the plate. So the loop needs to be cool, because if it's very hot, it can actually kill the bacteria or yeast that you put onto your loop, and you don't want them to be killed. So in this example, I have some E. coli on a solid medium. So now my loop is sterile and cool enough. I'll just check by touching it to the surface of the plate. It didn't sizzle, so it's ready to be used. So you want to collect a single colony from this plate and transfer it onto your new sterile agar plate.
Okay, just for you guys to understand, a single colony would be like one of the little round bacteria that you would see going on it. Don't worry, guys. As I said, this is will all be reiterated and explain to you guys if you guys do biology, but I just want you guys to know from now. And you guys can also do your own reading, or if you need help, I can always explain again.
So remember, when you labelled your plate, it was upside down, so you need to turn it now the right way up. If you feel uncomfortable when streaking, you can actually draw the streak lines as per your diagram onto the bottom of the plate. For this demonstration, I'm not going to do that because it will obstruct the view. So now I have my colony on my loop. I'm going to perform the primary streaks. So basically, you move your loop with the bacteria on it back and forth onto the surface of the agar. And that has to be done very gently so that you don't tear the agar. The agar is hard, but it is like a jelly substance and it can easily tear. So once you've transferred it, you need to flame your loop again to ensure that any excess bacteria is removed. Remember, the purpose of the streak plate technique is to dilute the cells. So we're removing any of the cells that might have been on our loop. So you need to make sure that you turn the plate so that you can perform your secondary set of streak lines. So they must intersect with the primary ones. So by turning the plate, that helps you to remember that you need to intersect them. So you need to...
Okay, we usually turn the plates at about a 45-90 degree angle. So that's how we usually
drag the loop across the surface of the agar, this time lifting it off at each of the strokes. And then replace the lid. And then again, you need to flame your loop. So it's really important when you're streaking as well that you make sure your plate isn't exposed to the air for very long. So after each set of streaks,
yeah, as you were saying, guys, it's important that you keep the lid not too far off the plate because you don't want to expose it to the air, any microbes that might be in the air as well. Also, don't also be breathing too much or talking too much when you're actually doing these techniques as well, because your mouth might actually cause some microbes to actually fall on the plate as well. So it's very important when you're doing that as well. Not when you're doing aseptic techniques, not to talk too much over the plates, not to be breathing too much over them. Try to work in a very sterile environment.
You should replace your lid very quickly. So once the lid is cool, you can touch it to the surface to make sure it's cool enough, and then turn the plate and intersect your secondary streak lines to create your tertiary streak lines. Replace the lid and then flame the loop for the final set of streak lines. So I've turned the plate. I'm going to check that my loop is cool, and I'm going to intersect the tertiary lines with the quaternary lines. 1, 2, 3. And now I'm going to flip the loop upside down, drag a vertical line through those lines, and then a zigzag into the middle of the plate so that I get my single isolated colonies. You need to flame your loop once you've completed the procedure to make sure you remove any bacteria that might be on there.
Okay, guys, so I just want to show you guys over how the plate usually looks after you finish the streaking. So let me just show you guys how it looks again for those who probably forgot. So So it usually look like this, guys. So you can at the right hand corner, the more brownish yellowish looking area, the one at the middle, you can actually tell is probably the streak that was done first. Okay, let me just annotate, use like an annotator or something. Yeah, I suppose you can annotate, right? Okay, so this right here is what she would have more likely streaked first, right? And then this would have been the second streak, and then this would have been the third streaking. So let me just quickly show you guys. Okay, so this is your agar plate. Let me just quickly show you guys. I'm sure you guys probably didn't exactly see what she was doing. So example, you use your inoculating loop and you pick like a single colony right off here. So you just use the loop and you just pick off a single colony right off. No, sorry, not right there, because that's not even a single colony. Right here, you take a single one of these. This is actually a colony. So the little circle that you're seeing, guys, this is what I'm actually calling a single colony. And these are actually just clumps of bacteria that you're seeing right here coming to here. So maybe this person, maybe I take my single colony from here, right? And then I do some streaking and then like this, and a streak right here, and a streak right here, and then I'm like, all right, that's a streak. And then I probably turn my plate like at a 45-degree angle, and then I turned my plate at a 45-degree angle, and remember I would flame my loop again, let it cool, and I would take just a little bit from right here, just a little from right here, and then I would streak again. Then I would flame my loop again, allow it to cool, and then take a little from right here, and then I would streak again. And that's how you get the single colonies, because as you isolate the medium, so I'm sure that you guys probably weren't really understanding or seeing clearly what's being done. And that's going to be the case for the most part when you're doing it you're not going to, because remember these are microorganisms, you're not going to exactly see. So you have to, make sure you're using the inoculating loop and streaking it, because the only thing you guys are going to actually be seeing when you're doing the streak plates is just the streak lines that you're going to be doing. It's only when you put it back into the fridge or where make it incubate over some time, you'll be able to see the organisms grow, guys. So this is it for the aseptic techniques. I hope you guys really understand what I was saying. I know it might have not been clear to everyone, but I hope you guys if you understand, I'll be sure to try to maybe explain it in another session if we have time, or maybe try to give you guys videos that you guys can look on to understand it a better way that maybe I didn't get to explain it to you guys. But you guys are supposed to have your virtual lab now, so we're going to go into that. But any questions before we go? Any? None? Nobody don't want to say anything?